What's up guys, welcome back to another YouTube video. The next few months I'll be covering paper 6 skills for IGCSE biology. Now this is an alternative to practical paper and I'm going to break down every single skill that you need to answer every single question. This is the one paper you can actually get a 40 out of 40 on. I'm going to make a series of videos on every skill that you need. And today we're going to start with the basics. We're going to talk all the variables. We're going to talk the testing that you need. Let's get started. Now variables. What are variables? Variables are factors that you control in an experiment. Now I'm going to share this file with you so you can see what's going on as we proceed in this video. Now an example of variables, you have time, you have concentration, you have volume, you are changing, you have color change, all these things you've seen in paper 6. Now, however, we need to talk about 3 types of variables that you need to know, which is independent, dependent, as well as control variable. Let's start with independent. Independent variable is a variable. We're going to look deeper into these types of variables by looking at questions. Now, dependent variable is a variable you are measuring and it's affected by changing the independent variable. Now, if we look at the same example above, now let's say you're changing the concentration of proteins and it's going to affect the color change. So your dependent variable in this situation would be color change. Now, control variables are variables that you keep constant so that it does not affect your experiment. So you have an accurate result. Now we're going to look at question and past papers. You can pause it and read it for a little bit and try to identify the variables yourself before you continue this video. Now I'm just going to read what we need to know. Potato cells take up methylene blue dye when the potato tissue is placed in the dye solution. If the dyed potatoes are placed in water, the dye will diffuse into the water. Now, this is a key, guys. I'm going to get a highlighter in your paper. Usually you're not allowed to use highlighters, so just get a pencil and try to underline whenever you're reading these investigations. Okay, now student investigated the effect of temperature on the diffusion of methylene blue. Now already you can see which one is your dependent, which one is your independent variable. Now they're telling you the effect of temperature. So essentially they're telling you if the temperature is high, if the temperature is low, how does the diffusion of methylene blue into your potato cells change? How is it affected by change in temperature, effect of temperature? So what you're changing is your temperature. So independent variable in this scenario is your temperature. Now dependent variable in this scenario, they're telling you, oh, what is the effect of temperature on the diffusion of methylene blue from dyed potato cells? So diffusion. So rate of diffusion, or that time taken for diffusion. So if I go a little deeper into the question, I didn't add it in here. So they're trying to measure the time taken for diffusion. Okay, so my dependent variable in this scenario is going to be time. Let's go into the next question. Now get your pencil or your highlighter. Always make sure you're highlighting what's important. Now students investigated the effect of temperature on the activity of amylase. Amylase is an enzyme that catalyzes the breakdown of starch to form reducing sugars, and then it describes the whole methodology to the experiment. I'm going to send this file like I said, so you can pause this video and try to read this experiment. What we're trying to do here is to identify an independent variable. This 1 mark is a giveaway, guys. They tell you in the beginning of this experiment and it tells us the effect. Whenever you see the word effect, you're trying to see, oh, that's what they're changing. In this scenario, they're changing your temperature. So I'm just going to say temperature. And if they ask me what's my dependent variable, if I look back down in this experiment, they're going to be recording the color. So the color is going to be my dependent variable if they're asking me. Now they're telling me what are the variables that are kept constant. This is my control variables. I'm keeping it constant to make sure that my result is not affected by anything else apart from temperature. So if I look at here, they tell me put 2 centimeter cubes of 2% amylase solution into 2 test tubes for cold water and hot water. Now, so you need to take same volume of amylase solution. So that's my first one. Okay, be as specific as you can. You don't have to say 2 centimeter cubes, you just need to say same, and that's essentially your keyword there. They also use the same concentration of amylase solution. Now, a lot of people try to use words amount, moles, these are very vague. Not amount, what type of amount? Volume or concentration, okay? So don't be very vague with your wording. Same concentration of amylase solution. I'm going to go back. I'm going to see, I'm trying to find another one. Now, obviously, you need to be reading the experiment. Now, I'm just trying to look through here. Now, it tells you leave both test tubes for 3 minutes. Okay, that's to allow the amylase solution to break down your starch to reducing sugars. So here what I'm going to say is wording. Remember the wording you need to be careful for. So we're going to say time the test tubes were left before, and if you look up here, after 3 minutes you put the amylase solution into your test tube C. So my last concept variable would be time the test tubes were left before amylase solution was added, and in the bracket for this one I'm going to write 3 minutes each. I need to be a little bit more specific for this one because it's not very easy to write it down. Now, another experiment. Now, a student investigated the effect of surface area on diffusion. Usually the first sentence is a straight away giveaway. So it tells you what your independent variables are just by the first sentence. Now take a pause and read it. Now we're going to see. State the independent variable and the dependent variable in the investigation described in 1B. Independent. There you go. 1 mark giveaway. Essentially, these are your 3 marks, okay? Literally in the first sentence it tells you effect. Remember, what did we say? Effect means independent. You're trying to identify which independent is. So they're changing the surface area and they are measuring the diffusion. And if we look a little closely, diffusion is, it is something they're measuring, but how are they measuring the diffusion? They measure the percentage of light absorbed by the methylene blue dye in the water. Okay, that's your dependent variable. That's what you're measuring. So if you're measuring, okay, these are keywords you can use to identify. That means that's your dependent variable. If it's an effect, that's your independent variable. So make sure you don't just immediately say your dependent variable is diffusion, because in this case, that's what's put in the first sentence. But as you go down and read the actual experiment, you can see that measuring the percentage of light that's absorbed by the methylene blue dye, which tells you the diffusion, which, is a way that you can measure diffusion rate. Okay, independent variable is surface area, and then your dependent variable is percentage of light absorbed by the methylene blue dye. Oops, 2 marks each, and I will be getting those 3 marks. Next up, we're talking chemical tests. Now, there is no short way out of this. You have to memorize them, and they're not that much. So now the first one we're going to look at is reducing sugars, a very generalized way of saying glucose. In a chemical test, whenever they ask you to describe one, you need to talk about the reagent, and you need to talk about your positive result, the color, and then that will give you your 2 marks. Now, the reagent in the scenario of reducing sugars is Benedict solution. Now, however, this is the one situation in reducing sugars only that you have an additional step in which you have to heat in water bath. I'm going to put equal solutions of volume and your Benedict solution, and then you're going to put in a water bath under a Bunsen burner. So 100 degrees Celsius. You don't have to be that specific. Just say heat in a water bath. The positive result is you're going to get brick red. Now, depending on the concentration, you could get green, yellow, anything but not blue. And your negative result, the color of Benedict is blue, so it just remains blue. So that means no reducing sugar in your sample. And then you get 3 marks. Perfect. Now, let's do a little question to practice this knowledge. A scientist investigated the nutritional content of fruit juice. Name the reagent that would be used to test for starch. We're going to look at this later, so we'll come back to this. State the name of the reagent that would be used to test for the fruit juice for reducing sugars. It is Benedict solution. Think of Benedict Cumberbatch. The next test we're looking at is a protein test. The reagent for this is biuret solution. It's a little similar, it has a B, and you could confuse it with Benedict for reducing sugar, but no, it is biuret. The positive result is purple, lilac, violet, anything purple, okay? Negative result, because biuret is blue, is going to remain blue. And that's your 2 marks, guys. That's the 3 marks. The reagent for the fat test. Fats, you put oil and water, they don't mix. So it's basically emulsification. So your reagent is going to be ethanol, which you're going to use to emulsify. And your positive result is white precipitate. And then you have negative result, it's just nothing's going to happen. Vitamin test. The reagent for vitamin test is DCPIP. You don't need to know what it stands for. Now the positive result for this is colorless solution, and the negative result for this is it remains white because DCPIP is a white solution. A starch test, the reagent you need to use is iodine solution. I'll actually add a table in this note so you can download them and paste the table next to your bed so that you can wake up and look at it every day. And anyways, the positive result for this is going to be blue blood. And because iodine solution is orange, brown sometimes, the negative result is going to be remains orange/brown. Normally in your exam, they never ask you what the negative result is, and you won't get a mark for knowing the negative results, but it's just a little extra information. Now that we know our tests, let's do a little exercise, obviously, to reinforce the knowledge. First one. Now, a scientist investigated the nutritional content of fruit juice. State the name of the reagent or solution that would be used to test for starch. Starch, always think, is iodine. State the name of the reagent that we use to test for reducing sugar. Benedict Cumberbatch. Benedict, okay? And then a sample of fruit was tested for protein. What is the positive result? You're gonna have purple or lilac. Don't make it complicated for yourself, but if you want to write fancy purple colors or shades of purple, write violet or lilac or whatever you prefer. Emulsification test is used to test for fat. What's the method? Describe. 2 marks. You're gonna use ethanol to emulsify/break down the fat. Show the examiner you know what you're talking about, okay? Don't just say ethanol. And then your positive result is white precipitate. Now, many people eat seaweed. Take the names of the reagent that can be used to test for seaweed, for protein, and vitamin C. Protein, remember, not Benedict, it's Biuret. And for vitamin C, we're using DCPIP. Cool name. So in this paper, I get 1, 2, 3. 3 plus 2 is 5. 5 plus 2 is 7. 7 marks, guys. So thank you guys for watching. I'll be sharing with you this notes and this paper so you can revise and look back at it. Thank you guys for watching this video. See you in another one.